This presentation is focused on acetaldehyde, the cumulative and most prevalent human carcinogen worldwide. The good news is that exposure to acetaldehyde can be markedly decreased at both population and individual level. The following presentation is broken up into 6 parts. Part 1 includes some key facts about upper digestive tract cancer epidemiology in different parts of the world. Part 2 focuses on some basic characteristics of acetaldehyde, for example, why acetaldehyde is the most widespread human carcinogen. Part 3 deals with outdated opinions of food authorities on acetaldehyde. Part 4 covers the metabolic pathways of acetaldehyde and provides the indisputable evidence for the causal relationship of acetaldehyde with upper digestive tract carcinogenesis in humans. Part 5 concentrates on the prevention of acetaldehyde-related cancers at population and individual level. And the last topic, in Part 6, presents a novel approach for the elimination of acetaldehyde in the oral cavity and stomach by slow-releasing L-cysteine, the Acetium formulations. Part 1. Key facts on upper digestive tract cancer epidemiology in different parts of the world. About 186,000 new cases of upper digestive tract cancers are diagnosed each year in 28 countries of the European Union, representing 7.1% of all cancers. These cancers have a particularly poor prognosis. The annual mortality is around 115,000, which is about 9% of all cancer deaths. Both the surgical and medical treatment of these cancers is challenging, making costs at a population level and individual suffering extremely high. It is generally agreed that the main focus regarding these cancers should be on their prevention. In the USA, the annual incidence of upper digestive tract cancers is about 76,000, representing 4.8% of all cancers. These cancers too have a particularly poor prognosis, and the annual mortality is about 36,000, which is 5.9% of total cancer deaths. Like in Europe, the surgical and medical treatment of these cancers is challenging, and so the same economic pressures at population level and extensive suffering at individual level are present. In the USA, it is also generally agreed that the focus should be turned towards cancer prevention. About 8,800 new cases of upper digestive tract cancers are diagnosed each year in Canada. This represents about 4.8% of all cancers. These cancers also have a particularly poor prognosis, and annual mortality is about 4,800, which is 6.5% of all cancer deaths. And, like Europe and the USA, the treatment of these cancers is challenging with extensive costs and suffering at both population and individual levels. Again, the consensus in Canada, like Europe and the USA, is to focus on cancer prevention. In Eastern Asian countries, upper digestive tract cancers form a colossal problem. In those countries, over 600,000 new cases, representing a quarter of all cancers, are diagnosed annually. Mortality from these cancers is over 440,000 per year, which is 25.1% of all cancer deaths. There is a particularly high incidence of these cancers caused by environmental and genetic factors. These include very high incidence of Helicobacter pylori infection, resulting in high incidence of atrophic gastritis. But another major factor is an increased exposure to carcinogenic acetaldehyde, which is secondary to genetic and dietary factors. Due to a gene mutation, 30 to 60% of the population are unable to eliminate acetaldehyde normally. Dietary factors include the regular consumption of pickled vegetables or other fermented products known to contain high acetaldehyde and elevated alcohol levels. Furthermore, Eastern Asian countries are characterized by increasing consumption of alcohol and tobacco. It's a common misconception that only heavy drinking and/or alcoholism increase cancer risk. However, a recent meta-analysis, including 222 studies, 92,000 alcohol consumers, and 60,000 teetotalers, clearly showed that even slight alcohol consumption representing one standard dose, which is equal to 10 grams of alcohol per day, significantly increases the risk of mouth, pharynx, esophagus, and breast cancer. It was concluded in 2004 that slight alcohol consumption caused 5,000 deaths from oral and pharyngeal cancer, 24,000 deaths from esophageal cancer, and 5,000 deaths from breast cancer. Part 2. In this section, we present the basic characteristics of acetaldehyde. For instance, why acetaldehyde is the most prevalent worldwide human carcinogen. A key to cancer prevention is the identification of specific etiological factors and carcinogenic compounds. Examples of widely known and specific human carcinogens, Group 1, are, for instance, asbestos, formaldehyde, benzene, tobacco, alcohol, and radon. The use of such known carcinogens is strictly regulated by internationally accepted laws. These directives have been shown to be very effective in cancer prevention. The International Agency for Research on Cancer has recently reclassified acetaldehyde as a Group 1 human carcinogen. A causal relationship between alcohol-associated acetaldehyde and upper digestive tract cancers has been consistently demonstrated in gene epidemiological and biochemical studies. Acetaldehyde is a key metabolite in alcohol production by fermentation, and also in the oxidation of alcohol via acetaldehyde to acetate in the human body. Acetaldehyde has an apple or orange flavor, its boiling point is close to room temperature, and it is easily water and lipid soluble. Therefore, it passes without difficulty through cell membranes. Acetaldehyde is cumulative and the most common human carcinogen. It is present in most alcoholic beverages, as well in foodstuffs produced or preserved by microbial fermentation. Acetaldehyde is widely used as an aromatic agent and food additive, and it is the most abundant carcinogen of tobacco smoke. It should be emphasized that ethanol itself as a molecule is not carcinogenic. Carcinogenicity associated with the use of alcoholic beverages is by and large mediated via acetaldehyde. This concerns free acetaldehyde present, sometimes in high concentrations, in many alcoholic beverages. Furthermore, ethanol from alcoholic beverages is metabolized locally in the upper digestive tract to acetaldehyde by normal microbial flora and mucosal cells. It should be emphasized that foodstuffs and beverages containing significant or high levels of alcohol and mutagenic concentrations of acetaldehyde expose the upper digestive tract mucosa by the same mechanisms to carcinogenic acetaldehyde as alcoholic beverages do. The ethanol concentrations of these so-called non-alcoholic foodstuffs and beverages may range from 0.5 to over 10%. However, in epidemiological studies, only the consumption of alcoholic beverages is systematically followed and recorded. This causes a major gap and bias in upper digestive tract cancer epidemiology. Part 3. This section looks at the food authorities' opinions towards acetaldehyde, which are in part outdated and in part contradictory. It was in 1985 that the International Agency for Research on Cancer concluded that acetaldehyde is carcinogenic to animals. In 1987, acetaldehyde was classified as possibly carcinogenic to humans, Group 2B, and in 1995 this was confirmed. In 2009, the International Agency for Research on Cancer reclassified acetaldehyde associated with consumption of alcoholic beverages as carcinogenic to humans, Group 1. This conclusion was supported since there is sufficient epidemiological evidence showing that humans who are unable to oxidize acetaldehyde to acetate have a substantially increased risk for developing alcohol-related cancers, in particular of the esophagus and the upper aerodigestive tract. In 1995, the Flavors and Extracts Manufacturers Association concluded that acetaldehyde is a generally regarded as safe product, and on this basis, manufacturers are permitted to produce or add acetaldehyde to beverages and foodstuffs without restrictions. The Joint Food and Agriculture Organization and WHO Expert Committee on Food Additives confirmed the generally regarded as safe status of acetaldehyde in 1998. However, conclusive scientific evidence for the local carcinogenicity of acetaldehyde in humans has accumulated since 1996, and so the generally regarded as safe status of acetaldehyde is not only outdated, but in sharp contrast with the IARC's reclassification of acetaldehyde as a Group 1 human carcinogen. The positive news is that the Scientific Committee on Consumer Safety recently reassessed the safety of acetaldehyde in cosmetic products. The re-evaluation, entitled Opinion on Acetaldehyde, was published in December 2012, and this document is a unanimous opinion of 12 independent scientific experts. Based on the IARC's reclassification of alcohol-associated acetaldehyde as a new human Group 1 carcinogen, the maximum concentration of acetaldehyde in the final finished cosmetic product was decreased from 25 to 5 mg per litre. Furthermore, the expert group concluded that acetaldehyde should not be intentionally used in mouthwashing products. The generally regarded as safe status of acetaldehyde violates the EC regulation of the European Parliament and Council, number 178 from 2002. According to Article 14 of the regulation, food, including alcoholic beverages, shall be deemed unsafe if it is considered to be injurious to health. Considerations should be paid to possible cumulative toxic effects that are characteristics of acetaldehyde exposure. Consideration should also be made to categories of consumers with special health sensitivities, such as individuals with ALDH2 deficiency, a genetically deficient ability to oxidize acetaldehyde, and those patients with acid-free stomachs, characterized by enhanced microbial production of acetaldehyde. In Article 14, it also states that information on the safety or unsafety of food should be provided in the product label, or by other means generally available to the consumer. Part 4. This section concerns acetaldehyde concentrations of alcoholic beverages, fruits, and foodstuffs. On this slide, we have some examples of acetaldehyde concentrations of various alcoholic beverages. We can see that Chinese rice spirits, Italian grappa, tequilas and cognacs exceed the upper limit of acetaldehyde permitted for cosmetic products from 5 to 136-fold. Equally high acetaldehyde concentrations are found in Calvados and sherries, whilst the lowest acetaldehyde concentrations are found in red and white wines and in vodkas. The low concentration of acetaldehyde in vodkas is due to very efficient distillation procedures during manufacture. One should also note the great variation in acetaldehyde concentrations within the different beverages shown. Accordingly, if acetaldehyde concentrations are detailed on the product labels, consumers can easily make their own informed choices. Here we can see some of the most important concluding remarks on acetaldehyde concentration with regards to alcoholic beverages. First of all, it should be noted that the data concerns free acetaldehyde that is very reactive and responsible for the carcinogenic potential of acetaldehyde. Free acetaldehyde can be measured most reliably by headspace gas chromatography. Acetaldehyde is a natural compound of alcohol fermentation processes, and different methods used in manufacturing may result in huge quantitative differences between various products. For instance, due to poor distillation methods, very high acetaldehyde concentrations are found in some beverages exceeding the mutagenic concentration, which is 25 micromoles or 1.1 mg per litre, by as much as 616-fold, and compared to the upper limit for cosmetic products, which is 5 mg per litre, by 136-fold. The good news is that in wine's low pH, most of acetaldehyde is bound to sulfites that inactivate the reactive carcinogenic CHO group of the acetaldehyde molecule. It has been argued that acetaldehyde in alcoholic beverages and/or yogurts cannot be so dangerous since apples and berries contain similar acetaldehyde levels. This is not, however, true. Finnish forest berries are free from acetaldehyde, and the mean acetaldehyde concentration of apples is over 600 times lower than in Chinese spirits, and 11 times lower than in Finnish yogurts. With regard to fruits, the highest acetaldehyde concentration, a maximum of 18.3 milligrams per litres, is found in overripe bananas. High free acetaldehyde concentrations in yogurt is intentional and aim to produce a good apple-type aroma in the end product. That's achieved using the fermentation process with two types of microbes, both of which are very good acetaldehyde producers. These microbes are various strains of Streptococcus thermophilus and Lactobacillus bulgariensis. Some orange soft drinks may also have high concentrations of acetaldehyde, perhaps due to the addition of acetaldehyde to them as an aroma agent. The good news is that there are lots of foodstuffs that are either free from acetaldehyde or contain it in only low concentrations. In summary, the highest acetaldehyde concentrations are found in overripe bananas, whilst Finnish forest berries do not contain free acetaldehyde at all. High concentrations of free acetaldehyde are found in yogurts too, and in some orange soft drinks. Acetaldehyde is produced in yogurts by microbes used in the fermentation process, but acetaldehyde may also be added to some soft drinks to improve their aroma. Very low acetaldehyde levels are found in most fresh vegetables and cheeses. However, pickled vegetables may contain high levels of acetaldehyde and low levels of alcohol, secondary to the traditional fermentation processes used for preservation. As a matter of fact, the high consumption of pickled vegetables has been shown to be a significant risk factor for stomach cancer. It has been claimed that yogurt cannot be dangerous because healthy apples have equivalent levels of acetaldehyde. This claim is not true, however, because the maximum acetaldehyde concentration of apples is only 1/7 of that found in yogurts. Part 5. The following slides illustrate the key metabolic pathways of acetaldehyde, providing unquestionable evidence for the causal relationship between acetaldehyde and upper digestive tract carcinogenesis in humans. Over 90% of ingested alcohol is metabolized in the liver through acetaldehyde to acetate. Acetaldehyde is metabolized in the liver so efficiently that measurable levels of acetaldehyde are not released from the liver into the bloodstream. Acetate is oxidized in the muscles and other peripheral tissues further to carbon dioxide and water. Thus, acetaldehyde has no systemic carcinogenicity. The key role of the liver, as the main organ for ethanol and acetaldehyde elimination, is more clearly illustrated on the right-hand side of this slide. Ethanol is oxidized in the liver through acetaldehyde to acetate. Acetate is released from the liver into the blood circulation, but not acetaldehyde. Acetaldehyde accumulates in saliva and gastric juice because a small amount, less than 1%, of ingested alcohol is metabolized to acetaldehyde locally by normal oral microflora and mucosal cells. In sharp contrast to the liver, oral microbes and mucosa have insufficient capacity to metabolize acetaldehyde further. Consequently, the highest acetaldehyde concentrations after alcohol intake are found in the saliva and gastric contents of an acid-free stomach. This is more clearly illustrated on the right-hand side of the slide. Ethanol is oxidized by oral microbes present in the mouth and an acid-free stomach, and by mucosal cells locally to acetaldehyde, which accumulates in the saliva and gastric juice. This slide illustrates the instant effect of a sip of an alcoholic beverage on salivary acetaldehyde. In this study, 5 milliliters of vodka or Calvados with an acetaldehyde concentration of 92 milligrams per liter was rinsed in the mouth for 5 seconds and then spat out. Saliva was collected at 2-minute intervals over the next 10 minutes. After the fast distribution of alcohol to the water phase of the oral cavity, some alcohol stayed in the saliva for the whole follow-up period of 10 minutes. This was associated with a marked local acetaldehyde production in the saliva, the blue columns after vodka, the light brown columns after Calvados. During the 5 minutes after sipping alcohol, salivary acetaldehyde concentrations stayed above the mutagenic level of 25 micromoles, or 1.1 milligram per litre, and, in some individuals, for up to 7 minutes. Sipping Calvados, with a high concentration of free acetaldehyde, resulted in a marked additional peak of acetaldehyde in saliva lasting for about 2 minutes, shown here by the light brown columns. This slide demonstrates the effect of 3 standard doses of alcohol on salivary acetaldehyde. In this study, human volunteers drank 3 doses, which were 0.5 grams per kilogram, of alcohol within 20 minutes, and afterwards their salivary acetaldehyde and ethanol concentrations were measured over 4 hours. In this experimental model, ethanol was diffused into the saliva from blood circulation, and acetaldehyde was locally formed from salivary ethanol by microbes and mucosa. In another experiment, the volunteers rinsed their mouth twice a day with 10 milliliters of 10% chlorhexidine solution containing 0 ethanol for 3 days. In both experiments, without chlorhexidine, represented by the blue columns, and with chlorhexidine, represented by the red columns, salivary acetaldehyde levels remained elevated for the entire observation period of 4 hours. Peak acetaldehyde levels clearly exceeded the in vitro mutagenic acetaldehyde concentration of 25 micromoles. Rinsing with chlorhexidine mouthwash significantly decreased both salivary acetaldehyde concentrations and the total number of oral microbes present in saliva samples. Many epidemiological studies and meta-analyses have uniformly shown that tobacco smoking is an independent and major risk factor for oral, pharyngeal, esophageal, and stomach cancers. About 30 cigarettes a day results in an 8-fold increased risk for esophageal cancer. Acetaldehyde is the most abundant carcinogenic compound of tobacco smoke. During smoking, acetaldehyde dissolves easily into the saliva and is subsequently distributed to the mucosal surfaces of the whole of the upper digestive tract. Alcohol and tobacco smoking have been shown to have a synergistic effect on the risk of upper digestive tract cancers. This old but very good study from France is still valid, and its results have been confirmed in many subsequent studies and meta-analyses. These subjects, drinking about 2 bottles of wine per day and smoking over 30 cigarettes per day, had a 150-fold increased risk for esophageal cancer as compared to the ones drinking moderately, 3 or less drinks per day, and smoking from 0 to 10 cigarettes per day. In accordance with epidemiological findings, smoking and alcohol drinking do have a synergistic effect on salivary acetaldehyde. In this study, smokers and non-smokers at first drank 0.8 grams per kilogram, or about 5 doses, of alcohol. Then, between 50 and 60 minutes later, smokers smoked 6 cigarettes at 20-minute intervals. In smokers, saliva samples were collected for acetaldehyde analysis before and after smoking, and twice during the smoking period. In the non-smokers, salivary acetaldehyde levels were measured at 20-minute intervals. During smoking, salivary acetaldehyde concentrations, represented here by the black spots, were more than 10-fold compared to the levels with alcohol only, the open circles. The area under the curve of salivary acetaldehyde of smokers was 7-fold compared to non-smokers, and the difference was highly significant with a p-value of less than 0.001. In order to be 100% certain about the causal relationship between acetaldehyde and cancer, thousands of human volunteers should be randomized to drink increasing doses of acetaldehyde or placebo daily. Both for practical and ethical reasons, this is not possible. Nature, however, provides such a unique human cancer model. A point mutation in a gene regulating the activity of the major enzyme, ALDH2, responsible for acetaldehyde elimination, has randomized tens of thousands of alcohol-drinking East Asians for decades to markedly increase local exposure to acetaldehyde. In moderate drinkers, the mutation is associated with a 2 to 5-fold, and heavy drinkers with an over 10-fold increased risk for oral, pharyngeal, esophageal, and gastric cancers. A comparable human model is not available for any of the other 113 human group 1 carcinogens. The origin of the point mutation in the ALDH2 gene has been suggested to be derived from an ancient Pai-Yui tribe, which was widely distributed among the southeast coast of China about 2,000 or 3,000 years ago. This evidence is based on current distributions of languages, culture, habits, and historical documents. Today, the gene mutation affects over 500 million individuals in China, Taiwan, Korea, and Japan, as well as individuals with origins in those countries. The main characteristics of ALDH2 deficiency are shown on this slide. In homozygotes, the ALDH2 enzyme is undetectable. These individuals are protected from alcoholism because of a severe flushing reaction associated with alcohol drinking. However, if they drink and smoke, they have a particularly high risk for alcohol-related cancers, up to 400-fold. In heterozygotes, less than half of the enzyme activity is detectable in most somatic cells. Thus, not only in liver cells, but also in, for example, gastric mucosal cells, and these subjects are characterized by less severe flushing reactions. They may become heavy drinkers and alcoholics, and most importantly, they have marked elevated acetaldehyde concentrations in their saliva and gastric juice after drinking alcohol. The enhanced local acetaldehyde exposure is associated with a marked increased upper digestive tract cancer risk. The striking effect of ALDH2 deficiency on salivary acetaldehyde concentrations after drinking of alcohol is demonstrated on this deficiency on gastric juice acetaldehyde concentrations. Chinese volunteers with ALDH2 deficiency of alcohol, or those with active enzyme per kilogram, ingested a moderate dose of alcohol of 0.5 grams per kilogram. Represents subjects salivary acetaldehyde concentrations of with normal active were followed over the next 4 hours, and the red line, as seen on this slide, salivary acetaldehyde levels of ALDH2 deficients, again with the black spots, according to the areas under the curve than in those with active deficiency, resulted in a 5.6-fold increase in the exposure of the ALDH2 to acetaldehyde. The additional salivary acetaldehyde appeared to originate from salivary glands. In accordance with the enhanced exposure of the upper digestive tract, the mean blood acetaldehyde concentration at 60 minutes of ALDH2 deficiency was only also increases the risk of upper digestive tract cancer. These findings have subsequently been confirmed in 2 studies. ALDH2 deficiency increases the risk cumulatively. More drinking means higher risk. On this slide we can see the breaking results of Yokoyama and his group from Japan from 1996 and 1998. These findings have been confirmed in tens of studies and meta-analyses. In heavy drinkers, ALDH2 deficiency resulted in about an 11-fold risk for mouth, pharynx, and larynx cancer as compared to those drinkers with the normal ALDH2 enzyme. The corresponding risk for esophageal cancer was about 13-fold, and 3.5-fold for stomach and colon cancers. With regard to acetaldehyde as a human carcinogen, it is possible to conclude that by and large all known risk factors for upper digestive tract cancer are associated with increased cumulative or synergistic local exposure to carcinogenic acetaldehyde. Genetic polymorphisms, most clearly ALDH2 deficiency, provide direct evidence for the local carcinogenic potential of acetaldehyde in the pathogenesis of upper digestive tract cancers. alcohol and tobacco smoking increase upper digestive tract cancer risk in a synergistic manner and have a cumulative effect on local acetaldehyde exposure via saliva and gastric juice. Furthermore, smoking and drinking are responsible for poor oral hygiene, which enhances the microbial acetaldehyde production from alcohol in the oral cavity. Stomachs with no acid, for example in atrophic gastritis, is the most important risk factor for stomach cancer. In a recent meta-analysis, the use of proton pump inhibitor drugs was also shown to increase gastric cancer risk. Many strains of Helicobacter pylori are also known to possess alcohol dehydrogenase enzymes, and these are able to produce acetaldehyde from alcohol. In East Asia, the frequent use of pickled vegetables has been shown to be associated with increased risk of stomach cancer. Part 6. The prevention of acetaldehyde-related cancers at a population and individual level are discussed in the following slides. With regard to acetaldehyde, there are many options that provide excellent possibilities for cancer prevention, both at population and individual level. Number 1 is the fact that the specificity of acetaldehyde as a human carcinogen is based on a unique human cancer model. It is also important that the wide presence of the acetaldehyde in our daily environment is rather well known. And thirdly, the main mechanisms regulating local acetaldehyde concentrations in the upper digestive tract are also well known. So how and why should we act at a population level? First of all, the generally regarded as safe status of acetaldehyde should be reassessed. It's no longer based on scientific evidence. Secondly, the use of acetaldehyde as a flavoring agent and as a food additive should be prohibited. Thirdly, necessary information about the hazards of acetaldehyde should be provided on the labels of beverages and foodstuffs for the consumer. The current situation violates Article 14 of the Food Law, Regulation EC number 178/2002, set out by the European Parliament and Council, and the generally accepted as low as reasonably achievable principle for carcinogens. Fourth, new processes for the production of acetaldehyde-free or low acetaldehyde alcoholic beverages and foods should be encouraged and supported. This is essential for the primary prevention of acetaldehyde-related upper digestive tract cancers. Fifth, health education on acetaldehyde should be provided for healthcare providers and for those in alcohol, food and tobacco industries, as well as for the consumer. This is crucial for both the primary and secondary prevention of acetaldehyde-related cancers. And how and why should we act at an individual level? The key message is that every one of us can decrease his or her acetaldehyde exposure considerably just by making our own informed decisions. The most effective way is to avoid or decrease one's tobacco and/or alcohol consumption. For example, by cutting alcohol and tobacco-related acetaldehyde exposure to half, it is possible to achieve a reduction in the risk of esophageal cancer by more than 20-fold. Another message is, don't drink to intoxication and opt for alcoholic beverages with low ethanol concentrations. Higher alcohol levels in saliva associate with higher salivary acetaldehyde levels. Thirdly, one should take care of oral hygiene, and microbial acetaldehyde production from alcohol decreases by up to 100%. Fourth, avoid alcoholic beverages with high levels of free acetaldehyde. They result in a short-term 10 to 30-fold increase in the local acetaldehyde exposure of the oral cavity. Fifth, choose acetaldehyde-free food and beverages, or select food and beverages with the lowest possible acetaldehyde concentration. There is new scientific evidence suggesting that fermented food, like pickled vegetables, and beverages, like dairy products, with high acetaldehyde concentrations significantly increase the risk for upper digestive tract cancer. Sixth, Pay special attention to established risk groups. Gene mutations such as ALDH2 deficiency or highly active alcohol dehydrogenase, acid-free stomachs caused by atrophic gastritis or drugs inhibiting gastric acid secretion, and chronic Helicobacter pylori infection all increase the local acetaldehyde production from alcohol in the upper digestive tract. Seventh, take advantage of novel, slow-releasing L-cysteine formulations patented and produced by Biohid Oyj, Helsinki, Finland. They effectively eliminate 60 to 90% of acetaldehyde derived from tobacco smoke and alcohol in the saliva and gastric juice. Part 7. The next 11 slides are focused on the elimination of acetaldehyde by novel, slow-releasing L-cysteine acetium formulations. L-cysteine is a semi-essential sulfur amino acid. Its mean daily intake ranges from 1 to 2 grams and may be up to 6 grams in individuals using high-protein diets. The essential characteristics of L-cysteine is that it binds covalently and non-enzymatically to acetaldehyde, and within seconds forms inactive methyltiazolidine carboxylic acid, or MTCA. MTCA is not absorbed from the intestine, and therefore it is excreted from the body via the fecal route. In the figure, the binding of free acetaldehyde with L-cysteine and its formulation of MTCA is demonstrated. In this study, human volunteers drank about 5 doses, or 0.8 grams per kilogram, of alcohol, and their salivary acetaldehyde levels were followed for 320 minutes. The experiment was carried out with placebo, in the blue columns, and with slow-releasing L-cysteine tablets, in the red columns. The tablets were fixed in the lower gingiva. The L-cysteine preparation contained 100 milligrams of L-cysteine. As shown on the slide, the slow-releasing L-cysteine tablet eliminated a mean of 59% of salivary acetaldehyde, and the effect of the tablet continued for the whole observation period of over 5 hours. In this study, the effect of a lozenge-type slow-releasing L-cysteine tablet on salivary acetaldehyde was examined during smoking. The volunteers smoked 1 cigarette, and all of the saliva for that time period was collected, and its acetaldehyde concentration was determined. As shown on the slide, 1.25 milligrams of L-cysteine in the tablet eliminated over 50% of the acetaldehyde from the saliva. With 2.5 milligrams of L-cysteine in the tablet, over 90% of salivary acetaldehyde was eliminated. In this placebo-controlled study, the effect of Acetium Lozenge on salivary acetaldehyde concentration after a sip of Grappa was tested. The Grappa had a very high acetaldehyde concentration, 235 milligrams per litre. Acetium Lozenge contained 3 milligrams of L-cysteine, or placebo. The volunteers dissolved 1 lozenge in their mouth for some minutes before the experiment, and thereafter they kept 5 ml of Grappa in their mouth for 5 seconds. Immediately after spitting out the Grappa, they took another placebo or L-cysteine containing lozenge. Saliva was collected at 5-minute intervals for 15 minutes. The blue line is the placebo, and the red line the Acetium Lozenge. As shown on the slide, the Acetium Lozenge eliminated a mean of 87.5% of salivary acetaldehyde, and the effect was highly significant with just this rather small number of volunteers, that is, n equals 3 for Acetium and 4 for placebo. The Acetium capsule is a novel innovation. From this capsule, L-cysteine is released slowly into the stomach, where acetaldehyde is formed. The indications for Acetium capsules are atrophic gastritis, use of drugs inhibiting gastric acid secretion, for example, proton pump inhibitors, surgically resected stomachs, and chronic Helicobacter pylori infection. Atrophic gastritis results in an acid-free or hypochlorhydric stomach. Its prevalence in developed countries ranges from 2.5 to 5%. The condition is most often symptom-free, or there are non-specific symptoms. Atrophic gastritis is often accompanied with vitamin B12, calcium, and zinc deficiencies, and the disease is detected most often by chance during gastroscopy. Nowadays, atrophic gastritis can be diagnosed from a simple blood test, and GastroPanel is produced and patented by Biohit Oyj in Helsinki, Finland, with a test that consists of the analysis of pepsinogen 1, pepsinogen 2, gastrin 17, and Helicobacter pylori antibodies. An acid-free stomach, known as achlorhydria or hypochlorhydria, is the most important risk factor for gastric cancer. In those patients with total atrophy of the gastric mucosa, the risk is up to 90-fold compared to those with a normal healthy stomach. Oral microbes are able to survive and multiply in acid-free stomachs. In the presence of any beverage or food containing alcohol, these microbes produce carcinogenic acetaldehyde locally. Also, glucose may serve as a source for acetaldehyde. Dark blue columns on this slide represent markedly elevated acetaldehyde levels in the gastric juice of achlorhydric patients after alcohol administration. Yellow columns represent gastric juice acetaldehyde levels of controls with normal acidic or healthy stomachs. On this slide, we can see an achlorhydric or acid-free stomach that is colonized by oral microbes, the red ovals. Furthermore, in the stomach there are slow-releasing L-cysteine capsules and 15% alcohol. In a placebo-controlled study, and under these conditions, L-cysteine, 200 milligrams, bound 60 to 70% of the acetaldehyde in the gastric juice, and the effect of the capsules lasted for the whole follow-up period of 40 minutes. In this Japanese and Finnish placebo-controlled study, we can see the effect of L-cysteine releasing Acetium capsule on the gastric juice acetaldehyde levels of 10 PPI-treated ALDH2 deficient patients. The blue line and column represent subjects with placebo, and red line and column represent subjects with Acetium. Hypochlorhydria was induced by treatment with rabeprazole, 10 milligrams twice a day for 7 days. Thereafter, the volunteers ingested 2 Acetium capsules, each containing 100 milligrams of slow-releasing L-cysteine. That was followed by an intragastric infusion of 15% alcohol, 0.5 grams per kilogram, which represents 3 standard doses of alcohol. We can see that Acetium eliminated 60% of the acetaldehyde formed from alcohol, and that the effect of Acetium continued for the whole observation period lasting 2 hours. In this Swedish-Finnish study, conducted in the University of Uppsala, patients with atrophic gastritis received 15% ethanol and randomly 200 milligrams of L-cysteine or placebo through a nasogastric tube in a single blinded fashion and on 2 separate days. 5 milliliters of gastric juice were drawn every 20 minutes for 4 hours for pH, acetaldehyde, ethanol, L-cysteine, and MTCA concentration studies. As shown in the figure on the right, Acetium eliminated 68% of gastric juice acetaldehyde, and that the effect of L-cysteine continued for up to 2 hours, as shown on the left. From 40 minutes to 100 minutes, gastric juice ethanol levels were below the official 2.8% limit for ethanol in alcoholic beverages. Nevertheless, acetaldehyde levels of gastric juice remain markedly elevated. This indicates that acetaldehyde production also takes place in the stomach in low ethanol concentrations, representing that of many alcohol-containing foodstuffs and so-called non-alcoholic beverages. The blue columns in this figure from the Uppsala study show that L-cysteine releasing slowly from Acetium capsules persists in the stomach for up to 3 hours. The red columns of the figure prove that acetaldehyde formed intragastrically from ethanol in the presence of L-cysteine is indeed converted to inactive and unabsorbable methyl thiazolidine carboxylic acid. The main conclusions of this presentation are shown in the last two slides, and they include the following key arguments. The carcinogenicity of acetaldehyde is based on a unique genetic human model. Free acetaldehyde is the most abundant worldwide human carcinogen. It's present in most alcoholic beverages and in many foodstuffs. Alcohol from beverages and foods is locally metabolized in the upper digestive tract to acetaldehyde. Acetaldehyde of tobacco smoke dissolves in the saliva and by this means is distributed to the oral cavity, the pharynx, esophagus, and stomach. Acetaldehyde exposure can be markedly decreased both at population and individual level. Slow-releasing L-cysteine from Acetium Lozenges, which contain 3 mg of L-cysteine, eliminates over 90% of salivary acetaldehyde during smoking and after alcohol intake. Slow-releasing L-cysteine from 2 Acetium capsules, each containing 100 mg of L-cysteine, eliminates 60 to 70% of gastric juice acetaldehyde after drinking alcohol or intake of any beverage or food containing ethanol. Acetium formulations, patented and produced by Biohit Oyj, Helsinki, Finland, are so far the only commercially available products for the local elimination of carcinogenic acetaldehyde in the oral cavity and stomach.